Hello, I'm Landon Warder, and I'm going to be talking about the work I've been doing on quantifying antimicrobial use and assessing the bacterial resistome on Canadian dairy farms and how that works towards an on-farm benchmarking system. I don't think it's news to anyone that antimicrobial resistance is a big issue in animal agriculture these days. There is plenty of room for there to be significant problems relating to human health and One Health, but the present issue today is really about treatment-resistant infections on dairy farms and how that affects the dairy cattle and their welfare and the economics of them as well. Uh, the adaptation to antimicrobial use and developing of antimicrobial resistance occurs when bacteria have replicative cycles in subinhibitory concentrations of antimicrobials, so they have a chance to reproduce and mitose in an environment that contains some of the antimicrobial, but not enough to prevent the mitosis or um, not enough to kill the bacteria. And this can lead to the evolutionary pressure to develop this resistance. So the combination of a benchmarking system and providing regular feedback can help guide farmers and veterinarians in their antimicrobial stewardship to help prevent these situations from arising and prevent the further emergence of antimicrobial resistance. Due to this increasing pressure from the antimicrobial use in uh, animal agriculture, there have been increasing regulations in Canada and around the world to try to encourage more antimicrobial stewardship and various methodologies. So since 2018, all medically important antimicrobials for use in animals require a prescription. Health Canada breaks these down into four categories of importance. Category 4 are considered low importance and are not medically important. Categories 1 are very high importance, so that's ceftiofur, fluoroquinolones. There are others as well, polymyxins, but ceftiofur and fluoroquinolones are the ones of main concern in dairy today. This maps pretty much onto the critically important list that the World Health Organization puts together. Category 2 are some very commonly used antimicrobials that are still considered high importance to human medicine. So that's penicillins and potentiated sulfonamides, as well as macrolides, very common groups. And then category 3 is considered moderate importance. Those are things like tetracyclines and phenycols. Further regulation came in 2019 with Quebec instituting these very stringent requirements around category 1 antimicrobials, requiring that not just that a prescription, but having some sort of reason that this particular infection warrants the treatment with category 1s. So beyond just the antimicrobials that are being used on the farm, we need to consider the bacteria and their communities and genetics that are being affected. So on in a microbial ecosystem, the microbiome of milk, there's always going to be certain bacteria that are always present that form the core of those microbial ecosystems. And in a healthy microbiome, they're quite even, meaning that there's not going to be too much of any one taxa. So that evenness is kind of a hallmark of a healthy microbial ecosystem. And when they're hit by an antimicrobial, this leads to a decrease in the evenness. Some bacteria become much more common than others because some are going to be much more greatly affected by the antimicrobial. And the microbiome, with becoming less even, becomes less diverse as well. So that becomes quite important when looking at not just the evenness, but the number of taxa that are present. That combination is the diversity. But once the antimicrobial is cleared from the system, these ecosystems have a tendency to go back to the state that they were in before, just due to the same inputs being present that will guide them back to the state that they were before. So you'll end up with a similar evenness and diversity as before. And there's always going to be resistance genes present, even to antimicrobials that aren't going to be used on the farm. These genes aren't just present in the situations where they're actually being used to fend off the microbes. They'll be floating around after use and sometimes before they're used as well. So the aims of this study were overall to develop and implement the tools for antimicrobial use and resistome benchmarking on Canadian dairy farms. The first section is evaluating the use of veterinary dispensing records as a data source for automated antimicrobial use quantification. Then the second section will be looking at how we can evaluate antimicrobial use on Canadian dairy farms, and particularly how it's changed from an earlier study between 2007 and 8 to later studies from 2017 to 2020. And then lastly, I'm going to look at the work I've done evaluating bulk tank milk as a medium for benchmarking the microbiome and resistome of milk, so we can look at it as a medium for providing feedback on these issues to the farmers. So the first part of this is going to be looking at the computer program I've developed to convert veterinary dispensing records into dose-based antimicrobial drug use rates. So we have records that show up like this as a spreadsheet and convert them into something like this, a ranked bar chart, to examine the antimicrobial use of each farm. So as I mentioned, with 2018, the regulations became much more stringent on how antimicrobials can be attained for use in animal agriculture. This led to a lot of consolidation, so you can't just pick them up from the TSC store. You need to get them through a veterinarian, and you can pick them up at either the veterinary pharmacy or your veterinarian or another pharmacy, but it's going to be only in a few places, and it requires a lot more stringent record keeping. So you're going to get the pharmacy name of the product, the amount of the product that is purchased, and the date of the sale. With uh, with measuring antimicrobials, you can measure the number of bottles that are bought, but that's not very useful for thinking about what's actually happening in regards to resistance. So you can also think about the mass of the antimicrobial, but again, the potency of the different antimicrobial compounds is going to vary a lot, so mass isn't a great measure either. So in this project, I just used dose-based metrics. The one unifying concept here is that they are thinking about the use in terms of its actual therapeutic impact. So one is the defined daily dose. That's telling you the number of days of treatment conferred by a specific amount of antimicrobial. Then there's defined course dose, which is the number of courses that are conferred by that number or by that amount of antimicrobial. And then that can be standardized by the amount of animal time exposed to produce a rate, in this case, an antimicrobial drug use rate. This equation is the overall formula that is used. So you have the number of the product in a specific format. So you can think Depasilin in a 250 ml bottle. Then you can have the format. So this case, that'd be 250 mils, and the concentration of the antimicrobial in that product. So penicillin and Depasilin. And that'll give you a mass when they're all multiplied together. You divide by a conversion factor. I use the conversion factors developed by Lynn Lardet and her group out of Montreal that is specific for the specific dose-based metric, so say daily dose, or defined daily dose. The active ingredient, so penicillin, and the route of administration, so Depasilin, that would be as an injectable. And then that can be added up for all the active ingredients that have been used on the farm, and you can divide by the amount of animals and the duration, so the animal time exposed, to give an antimicrobial drug use rate. So the primary objective here is to develop an automated system that'll convert these veterinary dispensing records to an ADOR DDD or DCD. And then I want to validate that computer program to ensure that it's reliable, and then evaluate the use of these veterinary dispensing records as a data source for automated antimicrobial use quantification systems in the future, and how suitable they are. So much of this was actually just developing the program. So the idea here is that it's going to determine what antimicrobial product is referenced in each sale in the spreadsheet, and then determine how much of each antimicrobial active ingredient was included in that sale, and then convert that the amount into a mass, and then that mass into a dose-based metric that's specific for an active ingredient in that route of administration, and then correct for the animal time exposed. So just to walk through this spreadsheet, or not spreadsheet, flowchart, you take the raw veterinary dispensing record and then some manual reformatting just to make sure that it's all set up in the spreadsheet correctly. And then using a thesaurus of the different names that clinics use for a product and a white list of products that are known to be not antimicrobials, that produces a standardized veterinary dispensing record, and that can produce the quantity of each product in a format size. And then a drug product database can be used to take that quantity and convert it into, or to figure out the active ingredients in each product. And that drug product database also contains the concentration for the products. So that'll give you the mass of antimicrobial active ingredient. Then taking the conversion factors from Millen-Lardet to produce the dose-based metrics. But that's just as a count. So we need to try to standardize by the amount of animal time exposed. We can figure out the duration of observation from the standardized veterinary dispensing record, since that'll include the first and last date, which over a long enough time period, that's going to be pretty close to the start and end of the observation period. Then you can get the number of animals either from survey or from farm records or from, say, a THI program. And putting those two together, you can get a population correction unit to give you the animal time exposed. And then combining that with the dose-based metric, you get the antimicrobial drug use rate. For validating, I looked at a direct comparison from 5 farms in Ontario. That's quite a small N. I would like a much larger sample size, but that was all that we could get for a direct comparison. But we also looked at 26 farms on PEI just to check how the program is actually working and look for errors. So for that direct comparison, when we're looking at overall use, the total use here, the correlation is fair, but it's not significant, partly just from the low power from having such a small sample size. But you can see that category 3 is the one that's the least correlated, just from a visual inspection, having one that's far off from the trend of the others. And when you remove the category 3, so you just look at the very high importance and the high importance antimicrobials, the categories 1 and 2, then the correlation becomes perfect as far as a spearman rank correlation, so a non-parametric method. This is just a sample size of 5, so I wouldn't take anything too strong away from this. But it's worth noting that there seems to be a bit of a worry around some of the moderate importance antimicrobials, and something that needs to be considered further in further trials. But when we're looking at the overall trend and the high importance and very high importance, the correlation is quite strong. Looking at the data from PEI, the ADUR DCD, so the courses per animal year, the rate was 1.46 DCD per animal year. And for the ADUR DDD, so that was 6.21 daily doses per animal year. I'll give some context for those numbers in the next slide, but those are quite reasonable. Here we have the ranked bar chart, so each bar here is a farm, and this red line is the median, and nothing's jumping out as being very strange. There's no extremely high use. It's a pretty steady trend. So there's nothing that's catching my eye as being an error. I went through personally and looked at 150 entries in the dispensing records and calculated them by hand. I found no errors in the computer program. Anytime there's a discrepancy between the computer program and my manual evaluation, it was because I had messed up. So the computer program was more reliable than I was. So I showed that we had about 1.46 DCD per animal year and about 6.21 DDD per animal year. These are some other comparable dairy AMU quantification efforts. The first two are Canadian. So Saney and Lardet, we'll learn more about them in the next section. But with Saney, this was an effort done in 2007 and 8, and they found 5.24 DDD per cow year using a garbage can audit, which again we'll learn about more in the next section. This is a bit lower than I found with my evaluation. They had a different method of dealing with drugs that contain more than one active ingredient, so a bit thrown off there. But they also used per cow year rather than animal years, so it's hard to say. But we can say for sure that the the difference between 5 and 6 DDD may be meaningful, but it's not something that is a clear sign of an error. It's not, we're not off by an order of magnitude. And then Lardet, we look at the DCD. Again, it is, or this one is much higher than I found, but again, we're looking at cow years, so it's not animal year. Otherwise, it was a much more comparable method of computing the antimicrobial use. It was using a garbage can audit instead of veterinary dispensing records, so there's going to be some discrepancy there. But again, we're off by a factor of about two or between 2 and 3, but it's not 10. So this is a bit of a worrying sign, but we're looking at cow year, which we're going to be looking at about half the herd there. So that will bring them much more in line. Might be worth worrying about, but it's unclear. With Stevens, this was in Flanders and Belgium. They found 7.2 or 7.59 DDD per cow year. Cow year again, so this is going to be higher than per animal year. So that brings it quite in line. And with the compost, they use animal year, which is quite nice for a direct comparison. It was with treatment records, which they tend to be unreliable, but with this effort, they put in a great deal of work to make sure that their records were accurate. And they line up pretty much 1-to-1 for the DDD per animal year. So that's a good sign. And that's the most recent of these. It was in the US and looking at very large farms. So not directly comparable, but it's a good sign. And with Reading, this was with survey data, which is the least reliable, but the use rate for courses was very similar and used animal years, so more comparable. So overall, my takeaway is there might be some reason to worry about this, but the overall trend is the numbers it's producing are quite in line with established values. So it's not throwing up any major red flags for me. The challenges with this effort were about primarily about developing the thesaurus and safe list, because that meant that every time we entered new records, I had to update these files to produce the correct standardized veterinary dispensing records. I know the group has gone on to use some machine learning and AI to try to streamline the process to make it much more efficient, and apparently it's been working quite well. So it's good to hear that they're making some progress, but there is still caveats with using machine learning. And then there's also the manual conversion. We're getting these VDRs and anything from a well-formatted Excel sheet to handwritten notes. So there's still challenges there with natural language processing, but there's certainly things that can be done about those. I think the easiest one is integrating with the clinic management softwares and just getting the data straight from the computer. And then we also want to be working on feedback mechanisms so that we can close the loop here. So we can get the data back to farmers and veterinarians in the way that can help guide them in their antimicrobial stewardship most effectively. The next section is going to be looking at quantifying antimicrobial use on dairy farms using garbage can audits. So you can see some pictures of the garbage can audits, but I'll get into a bit more detail in a second. So there were a lot of changes in antimicrobial attitudes from the first major study of antimicrobial use in Canadian dairy farms to the present day. So the first one was done by Saney et al. in 2007 and 8, and they had sites across Canada. At that time, Radistitz, the main textbook for a lot of Canadian veterinarians, it recommended blanket dry cow therapy for all farms. It was the standard, and it only makes a passing reference to antimicrobial resistance at the time, and that's certainly not the case anymore. Selective dry cow therapy has certainly become more, might not be recommended in all cases, but certainly more encouraged. And there were a lot of rules and recommendations that changed from World Health Organization, Health Canada, Government of Quebec, and several other governments as well to put more emphasis on antimicrobial stewardship. So the objective from this study was to assess antimicrobial use in Canadian dairy farms, and then to compare between antimicrobial use in 2007 and 8 to 2017 to 2020 using garbage can audits, and then to see how these changes have affected antimicrobial use. So with the garbage can audit, a receptacle, like you can see these orange buckets, they're placed on the farm in a convenient location, and the staff at the farm are told to throw all of their medications once they're empty into that garbage can. Whether it's an antimicrobial or not, you throw everything into that can, and then a technician that comes by later and lays them out, as you can see here, on a very large farm, and we'll count everything in the garbage can and record it so that we know how many tubes of Spectromask or whatever that is, can't quite tell, were used in that time. And then you can use that data from the same calculations that were used in the previous study. So for the efforts that were most recently done in 2019 and 2020, which the study is the first to report on that quantification effort. There were sentinel sites set up in the Fraser Valley in BC, southwest of Calgary in Alberta, the London Middlesex region of Ontario, the Montérégie in Quebec, and in Nova Scotia. So by combining that effort with a study that was published by Alain Lardet on data from Quebec and the Montérégie, we could then make a comparison to a SANI study, which included measurement from all those areas except for BC. And you can make comparisons from the first time point, 2007-8, to 2017 to 2020. So in the most recent study in 2019-2020, we found similar usage to what we found in the previous study. With this is per 100 cow years, or 100 cow years. So 6.23 or 623 daily doses per 100 cow years, and 117 defined course doses per 100 cow years. And you can see the breakdown here for all of the categories of medically important antimicrobials. But the real question here is, how did use change between the first study by Saini et al. And the later studies? So this is using predictive margins from a fixed effect multivariable regression. So we have the natural logarithm of DDD per 100 animal years on the y-axis, and then for each of the 4 regions plus Canada overall, that we had data from both time points. We can see that there is a significant drop. These are all with 95% confidence intervals. So you can see that drop in each region was quite significant between the 2 time points. And overall, it decreased from 1,218 DDD per 100 animal years to 646 DDD per 100 animal years. I talked about Saini in the previous section. and the number that I used for their DDD per 100 animal years was much lower than this, but that was because in Saini's study they treated products that contain more than one antimicrobial active ingredient as just having one antimicrobial active ingredient. In this meta-analysis, I was able to get the raw data and run it through my pipeline so we could handle each antimicrobial active ingredient separately, and it about doubles their DDD per 100 animal years. I guess it was actually cow years here. So here we're just looking at ceftiofur, which is a bit of an exception. So in the previous one, you can see that there's a significant drop between the time points in all of the regions. And this trend is seen in pretty much all of the categories of use and all the chemical classes. But ceftiofur is a bit of an exception here. We see a very small increase in usage, actually, and you can see the confidence intervals overlap significantly. Which this is a concern because ceftiofur is the most used category 1 in usage in Canadian dairy farms. This is the one that we care about the most. But the big thing here is that we don't see a change in the usage much, but also in 2008, we got Spectrumast on the market. This was the first ceftiofur product for use as an intramammary product. So that introduced a whole new route of administration. So this is a real thing that there wasn't actually much of a change in use. And maybe that's an issue to do with validity, but the thing that makes the most sense to me is just that any reduction in usage in the systemic treatments from increases in antimicrobial stewardship stringency were probably offset by just having this entirely new product that introduces a new way of usage, which that's still a concern. It's still a major way that we can get resistant infections to be developed, but it is certainly an explanation that can help guide antimicrobial stewardship in the future. One other thing was there was one farm that was a massive outlier in this study, and in a lot of the DDD calculations, it would be extremely high leverage and could influence model building. But uh, all of that extreme usage by DDD came from 1/2 bag of chlorotetracycline, and it was a feed premix. And it's meant for continuous feeding for prevention, which is just a very different usage than, say, like boluses as an oral product, and it's just a very different way of usage. And it's hard to say what that even means for treatment because preventing infections is not really encouraged anymore. And if you're trying to use it as a growth promoter, that's just entirely not allowed at this point. So it really throws off a lot of these calculations and makes it hard to judge what this means from a therapeutic endpoint for the DDD, since you're not treating anything per se, you're trying to prevent something. And it's something that's supposed to be ongoing all the time. It's prevention. So, like, what's a course there, and what is a DDD there? So it really makes interpretation hard, and in a lot of the analysis, I had to exclude it because it's such a high leverage one where the interpretation becomes quite muddled. The third part of this project is characterizing the microbiota and resistome of bulk tank milk samples on PEI. So bulk tank milk is pretty interesting surveillance medium because it's readily available. It gets collected every couple days, and it's easy for the person doing the collection to just take a sample out of it. It's regularly done for some diseases, and it contains contributions from the majority of animals on the farm. But those animals don't contribute equally, and there are a lot of biochemical challenges of milk that can interfere with some of the biotechnical processes that we might want to use for some of this uh, biological stuff. So you can often do things like culture for determining the bacteria, but some cultures for some bacteria aren't able to be cultured. They're viable but not cultural, VNC, and for whatever reason, they won't be able to be grown in certain laboratory conditions. So they can instead be determined through DNA sequencing and looking for their genes in the sample. And even though these bacteria can't be cultured, that doesn't stop them from carrying antimicrobial resistance genes and spreading those genes to other bacteria, including pathogens. So they're very important that we can actually monitor them. So the objective of this section is to investigate the microbiota and resistome of the BTM, bulk tank milk. And we also want to determine any differences in the microbiotic composition between the cream and pellet fractions of the milk. And then we also want to investigate the feasibility of using bulk tank milk for target capture resistomics. So the bulk tank milk samples were collected from only 12 farms on PEI. This is, again, fairly small sample size, but this is a pilot to mostly look at the use of these biotechnical processes. 9 of the farms were freestall and 3 were tie stall. The milk is centrifuged, and that produces the cream fraction floating on top and then a cellular pellet at the very bottom. And the V4 hypervariable bacterial 16S rRNA gene, so the DNA that codes for this RNA, was amplified. This spinning piece of macromolecule sitting next to these points is the 16S rRNA that gets coded for. And then that can identify which bacteria are present. And then these amplicons can be sequenced with the Illumina HiSeq 2500 to figure out which bacteria are being represented by each one of these different 16S RNA genes. For the resistomics, there were 7 cycles on the PCR just to increase the DNA bulk, and then biotinylated cRNA baits for resistance genes, so looking for known resistance genes. And there's this biotin here is put onto the onto these markers that are looking for these genes, and then using the protein streptavadin that's attached to beads that binds biotin very strongly, then they can be concentrated out, and then 16 or 13 more cycles of PCR increases the bulk of just those resistance genes. So from the bacterial side, or from the microbiotic side, in the cream and pellet, here we have box plots of the main phyla, and we see the same sort of patterns. So firmicutes were the most common, followed by Bacteroides or Bacteroidetes, and Actinobacteria and Proteobacteria. So very commonly known, or very commonly seen bacteria, and we see the same ordering of these different phyla and their relative abundance across the cream and pellet. So it seems like they're quite similar between the cream and pellet, and depending on what fraction is easier for researchers to use for their specific application, seems like cream and pellet are pretty similarly useful for use in these microbiotic instances, though an absence of evidence isn't evidence of absence. There's nothing raising red flags here. When it comes to the antimicrobial gene or resistance gene reads, the counts varied a lot. You can see here that in the bulk tanks 2, 3, and 12, there's very, very few resistance genes. Very, very few being less than 100 after all those cycles once they were counted. But You can also see in some of these, they're well over 2,500, pushing 3,000. So there's a lot of variation in the uh, not just the number of counts, but the number of genes, but the number of times those genes are found in their relative abundances, which indicates that there's a lot of difference between farms and their um, their potential for not just having genetic resistance present, but having phenotypic resistance, as these genes can find their way into the genomes of pathogens. It was also interesting to look at, between those 9 freestall and 3 tie stall, about how different the diversity is. And by faith phylogenetic diversity, the diversity was much higher in tie stalls and freestalls, which is kind of an interesting thing to see. Um, this was significant on multivariable model, and can largely be explained by, when you have cattle interacting with each other all the time, they end up sharing a lot of their microbes, versus with tie stalls, they can have a bit more individuality to their microbiomes, which can um, start presenting itself in the milk, since you have a bunch of very different microbiomes contributing to the same sample. So it was interesting to see that there was no difference between the cream and pellet and the microbiotic diversity, um, or none that we could find. Again, not there's no there was an evidence of or an absence of evidence, not evidence of absence. But it certainly seems like it's worth moving forward with either the cream or pellet, as makes sense for the study, for future studies. Uh, and again, this is just a pilot, so it's just to establish a groundwork for these. On the resistomic side, the target capture techniques used here with the biotin and streptozan were able to find resistance genes in every sample, which is a great sign. And furthermore, there was a large degree of variation in the read counts, and this may be noise, or this may be an ability to detect, even, or to amplify the variation, so that we can have a higher empirical sensitivity for looking for differences in regards to different factors that may be affecting the resistance, like antimicrobial use. So there's more work that needs to be done here, but it certainly shows a lot of promise, and doesn't seem like there was anything about the milk or the bulk tank milk that was preventing the technique from working. So now, to put all these pieces together, we looked at 2 different ways of measuring antimicrobial use on dairy farms. So with VDRs, veterinary dispensing records, they're measuring the appearance rate of antimicrobials on the farm, and they're automatable, I was able to show. And they seem quite reliable. There is going to be more that needs to be done to make sure that each implementation is done well and isn't having any unknown errors, but there seems to be quite a lot of ability there for them to be automated. And then from the GCAs, they're measuring the disappearance rate, and they're easier to set up, but they are also have been proven over a couple decades now that they are quite useful for measuring antimicrobial use on dairy farms. So the other part of this is looking at how we can use this data to help guide antimicrobial stewardship. So I've whipped up a visualization, interactive visualization tool. So if a specific farm wants to understand their usage, they can go through and see their farm highlighted in comparison to their peers. So this is sort of what the benchmarking is. You can give them a distribution to understand, and then see where they compare. And you can use the different ways of measuring. You can break down by category, class, or root, and help give them an idea of where they might be above or below their peers, to help guide their stewardship efforts in the future. For the bulk tank milk portion, we want to lump that in with the stewardship to understand how these work together. And it was interesting to see that the free stalls had lower diversity in the bulk tank milk, and the sharing of the microbes that I was positing as the reason for this might be why, or might be playing a role, as we could have the sharing of not just the microbes, but the antimicrobial resistance genes spreading around the farm too. And the use of these bulk tank milk as a surveillance mechanism might be a good way of exploring that as a benchmarking tool or a surveillance mechanism for understanding how these resistance genes are moving around on farms. Overall, there were some limitations to the study or these studies. So, with the automated VDR processing, there's insufficient validation there. I only had 5 farms where I was able to do direct comparisons. So, it's going to be important if this is expanded on to have more direct comparisons with known methods like GCAs. With the change in what we saw in the GCA between 2007 and 8 and 2017 to 2020, the biggest concern I have there is with sampling bias with respect to external validity. The sampling procedures were very similar in Saini's study versus Lardet in the most recent one, but these are looking for farms that are willing to participate in these scientific processes that are going to examine their antimicrobial use. They are farms that were involved in dairy herd improvement programs. These aren't necessarily representative of farming in dairy in Canada as a whole. So the external validity is a little more questionable there. From the bulk tank milk, and insufficient power, having too small of a sample size, there are always challenges with these. The main concern here was to make sure that pellet and cream are useful, um, both for looking at the microbiome and for making sure the target capture can work on bulk tank milk. And I think there was enough power to demonstrate that there were no issues there that we could find, and enough to move forward with those as techniques in larger studies. But for takeaways like the free stall versus tie stall, that is preliminary, and I would want larger sample sizes to take that as a ground truth. Future directions, ongoing antimicrobial use quantification, I think, is the biggest thing, particularly in Canada. Uh, this is going to require increases in practicality, but also making sure that we're able to monitor trends in antimicrobial use and providing feedback for farms so that this isn't just something that happens every decade or two. It's something that farms can always have feedback on, because these farms have shown that they want to improve their antimicrobial use because they've really improved over that time, but they weren't even getting feedback. So it's nice to be able to show them how they're doing over time and let them see that they're doing a good job. And then we also want to be looking at the bulk tank milk antimicrobial resistance genes and how those change with changes in antimicrobial use in the herd to see how good of a surveillance mechanism it is and how useful that is as a benchmarking tool to start understanding how farms are comparing to each other. So, in conclusion, the VDRs have been seen to be a good source for automated antimicrobial use quantification. They worked for the first part of this project, so we know that they're good and worth exploring in the future. We've seen that Canadian dairy farms have reduced their antimicrobial use since 2007 and 8 to 2017, 2020, even without feedback. So it's going to be important to see where that goes from now, but we can take that away that they have improved. And we can also see that bulk tank milk is reasonable medium for target capture resistomics, and the pellet and cream fractions of the milk are both a very similar microbiota, and their use should be based on practicalities rather than on which one is seen to be a more reflective of the milk. I want to acknowledge my funders, NSERC, Dairy Farmers of Canada, Zoetis, and the Public Health Agency of Canada. That's all for me, and I thank you all for watching this presentation and listening and understanding my work. I appreciate being able to talk about it. Thank you. Bye.